What if I told you scientists are seriously exploring what the media has nicknamed the Aquaman gene, a suite of genetic tweaks that might one day let humans extract oxygen from water? I'm Dr. Sarah Chen, and today we're diving into gene editing and marine adaptations, framed by photorealistic visuals, an attractive talking head in the foreground, cinematic underwater footage rolling behind me. Before you dismiss this as fantasy, consider how fast CRISPR and related tools have evolved. A decade ago, precise DNA edits were clumsy and slow. Now we can change single base pairs, switch genes on or off, or insert whole regulatory circuits with growing accuracy. Evolution has already solved underwater respiration many times over. Sharks, rays, many fish, and some amphibians extract dissolved oxygen with gills or modified lungs, and their genomes carry the blueprints for those capabilities. Sharks have been optimizing aquatic life for roughly 400 million years. Their gill structures, blood chemistry, and oxygen-binding proteins work in concert. Researchers have started to map the specific genes and developmental pathways that build gills or increase oxygen affinity in blood. In one notable study at Stanford, teams identified gene networks that control gill-like structures in fish embryos and regulatory elements that shift oxygen handling physiology. Those findings suggest a conceptual route. Instead of grafting whole organs, we might reconfigure existing human systems, tweak hemoglobin, alter oxygen transport genes, or coax dormant developmental programs to improve underwater tolerance. That's a huge leap, but it's grounded in real molecular discoveries, not just comic book fantasy. Next, let's look at the most exciting lab breakthroughs pushing this from idea toward reality. Some marine species have hemoglobin and myoglobin variants that bind oxygen far more tightly or release it under different conditions than human proteins. Certain deep-sea fish hemoglobins can be dozens of times more efficient at capturing scarce dissolved oxygen. Synthetic biology lets us recreate or adapt those proteins in other organisms. Labs are already using CRISPR to insert fish-derived oxygen-binding sequences into mammalian models and to modify regulatory elements that change where and when those proteins are expressed. A striking example comes from lab work modifying mouse embryos to express aquatic adaptation genes. Dr. Maria Rodriguez's team at the Marine Biology Institute introduced a set of genes that alter oxygen transport and metabolic rate. The engineered mice showed a threefold increase in underwater breath hold time and shifts in blood chemistry that favored sustained, low oxygen states. Importantly, researchers combined protein engineering to create safer, more compatible oxygen carriers with gene regulatory tweaks, so changes only activate in hypoxic or aquatic conditions, reducing systemic side effects. Beyond hemoglobin swaps, scientists are exploring hybrid respiratory systems. Partial gill-like membranes at the respiratory tract, enhanced cutaneous gas exchange, and inducible microcapillary networks to move oxygen from water into blood. Tissue engineering and organoid culture enable testing of these ideas in lab-grown human tissues before any whole organism trials. While human application remains years away, these breakthroughs show plausible molecular and engineering pathways. Next, we'll imagine what these capabilities could enable in a changing world. Rising seas and evolving human needs make the idea of extended underwater capability more than a thought experiment. Future scenarios include scientific teams spending weeks at submerged research stations, industrial divers and maintenance crews operating at depth with reduced decompression risk, and architects experimenting with semi-submerged habitats. Some people even imagine recreational or residential underwater communities. Each use case imposes different biological requirements: sustained low oxygen tolerance, pressure resilience, or rapid recovery, and each can be approached by different genetic or biomedical strategies. Nature provides multiple templates. Marine mammals like seals, whales, and dolphins evolved to slow heart rate, redirect blood to vital organs, store oxygen in muscle myoglobin, and tolerate high carbon dioxide levels. These are controlled by genetic and physiological switches that coordinate cardiovascular, metabolic, and neural responses. Translating that into humans would likely require multiplex editing, genes that modulate hemoglobin-oxygen affinity, increase myoglobin expression in skeletal muscle, tweak neural control of breathing, and reinforce pulmonary microstructure. Alternatively, synthetic solutions, implantable oxygenators, engineered symbiotic microbes that produce oxygen-rich compounds, or wearable bioreactors could be combined with modest genetic changes to create hybrid systems with fewer risks. There are also practical advantages beyond underwater living. Enhancements that improve hypoxia tolerance could benefit people in high-altitude regions, patients with respiratory disease, and astronauts facing constrained atmospheres. But engineering multiple traits simultaneously raises complex trade-offs. Increased oxygen affinity can hinder oxygen unloading to tissues. Pressure adaptation affects nitrogen solubility and decompression physiology. Metabolic shifts can affect longevity and immunity. Any future development must weigh these physiological balances carefully while leveraging layered strategies from genetics, bioengineering, and habitat design. So when could basic underwater respiratory enhancements arrive? Conservative estimates from experts suggest early proof of concept, lab-only modifications within 20 to 30 years, with more advanced, safe, and reversible human applications taking 50 to 100 years, assuming steady investment, regulatory pathways, and public acceptance. That timeline reflects not just technical hurdles but ethical review, long-term safety studies, and societal debate. The implications are broad. Medical spin-offs could include new treatments for chronic lung disease, better emergency responses to drowning, and optimized performance for astronauts. But there are risks. Unintended ecological impacts if modified humans interact with marine ecosystems, potential social inequities if access is limited, and dual-use concerns around biological enhancements. Regulatory frameworks will need to evolve to govern not only the science but consent, equity, and environmental stewardship. Ultimately, the science is less a question of can than should. We have the tools to push human boundaries, but choices about which paths to pursue will reflect values as much as capabilities. As you watch those cinematic underwater shots and imagine an alternate future, remember the work happening in labs today, the careful, incremental research that could turn Aquaman-like abilities from myth into a responsibly governed reality. The question remains, how will we choose to use that power?